Inhibition in the central nervous system. Inhibition is prevention, decrease of or a stop of any activity in the body. So generally speaking, the first time inhibition was demonstrated in physiological experiment by an experiment that was conducted by scientists, two brothers Webers, who demonstrated that the stimulation of vagus nerve inhibited the cardiac activity. The inhibition in the central nervous system is a local process. It is an active process, and it is a result of interaction of two or more excitations. So inhibition does not propagate in the central nervous system. It is always a local process. Classification of inhibition in the central nervous system. In accordance with localization, inhibition may be classified into the presynaptic and postsynaptic. Presynaptic inhibition. So we may imagine that there is a synapse that conducts excitation. Receptors. And on the presynaptic membrane we have another synapse here that releases, for example, GABA, inhibitory neurotransmitter. So this inhibitory neurotransmitter hyperpolarizes the presynaptic membrane of the first synapse, and therefore when the second synapse is activated, the excitation will not pass through the first synapse. So the second excitation will be called inhibiting, and the first excitation will be inhibited. Interaction of two excitations will result in inhibition of the first excitation. In this case, we have presynaptic inhibition. Postsynaptic inhibition. We will draw the first synapse, receptors. This synapse conducts excitation from neuron 1 to neuron 2. And on the postsynaptic membrane, we see the second synapse. And the second synapse again may release GABA, inhibitory transmitter. This GABA will hyperpolarize the postsynaptic membrane of the first synapse also, and the excitation from neuron 1 to neuron 2 will not be able to pass through the synaptic cleft, because the sufficient level of depolarization of postsynaptic membrane will not be reached due to the hyperpolarization and inhibitory postsynaptic potential from the second synapse, and the level of depolarization will not reach the critical or firing level in the second neuron on the postsynaptic membrane. This is an example of the postsynaptic inhibition. Another classification of inhibition may be based on the mechanisms of the inhibition. We may distinguish the inhibition due to the hyperpolarization. For example, we just discussed the inhibition as a consequence of release of GABA or glycine that produce hyperpolarization of the postsynaptic membrane. Also, we may distinguish the inhibition due to the pessimal process. We already discussed the pessimum in the case of the tetanus muscle contraction. So the very high frequency of stimulation will inhibit the nerve centers also. Such inhibition may be the mechanism of the inhibition of vasoconstrictor center in the medulla oblongata due to the high frequency of stimulation of baroreceptors in the carotid artery. It will be discussed further. The third classification of inhibition may be in accordance with the cellular mechanisms due to the specific interaction of neurons that will result in the inhibition. The first mechanism is recurrent inhibition. We will draw a neuron, the body, an axon, and a branch of an axon, as we call it, collateral. So we then can draw the synapse, synaptic cleft, and the second neuron. The second neuron that gets excitation from the first one will generate an action potential that will propagate along an axon, and it will come back, it will return to the body of the first neuron, and this second neuron will release, for example, GABA or glycine, and will inhibit the first neuron. So when the first neuron generates action potentials, they will propagate in the nervous system, and they will also return to the first neuron and will inhibit this first neuron. So the significance of this inhibition is to prevent overexcitation in the central nervous system. Such feedback, negative feedback, will prevent overexcitation and excitotoxicity in the nervous system. Excitotoxicity is the process of cellular death due to the over excitation. Another cellular mechanism of inhibition is called lateral inhibition. So we may draw one neuron and in the vicinity the second neuron. So when the first neuron is excited, the excitation propagates along an axon, and an axon produces the collateral. This collateral, this branch, conducts action potentials to the body of the third neuron that will release GABA or glycine on the body of the neighbor of the second neuron. So, in that case, if the first neuron is excited, it will inhibit simultaneously its neighbors, and such inhibition is found in the receptor fields, for example, in the retina, in an eye, where when one photoreceptor is excited, rod or cone, then the surrounding receptors will be inhibited. Such inhibition is useful, and it increases the discrimination of an object. increases the contrast of analyzer sensation ability. Another inhibition that is important to remember is called reciprocal inhibition. In the case of reciprocal inhibition, we have the interaction between two nerve centers that are responsible for opposite effects on the body. For example, in the medulla oblongata, in the respiration center, we have two groups of neurons. Neurons that activate inspiration and the neurons that activate expiration. Of course, those functions exclude one another. In that case, if the inspiration center is activated, it reciprocally inhibits the center of expiration, and vice versa. So a person cannot inspire and expire simultaneously. Such interaction may also be found between the centers that activate flexors and extensors, muscle groups that do activate different movements of limbs and should exclude one another, or between the center of hunger and center of satiety in the hypothalamus, for example. A person cannot feel satiety and hunger simultaneously. Reciprocal inhibition. Another classification of inhibition may be based on the parts of the central nervous system that may be activated and may lead to the difference to two different examples of inhibition. The first inhibition that we should remember is called Sechenov inhibition. We will describe the Sechenov experiment. He used frog, and he did a surgical operation to open the access to the frog's thalamus. So we will draw the frog and we will show the thalamus, the part of the brain. Thalamus is a brain structure. And then he put the frog's leg into the glass with an acid, hydrochloric acid for example. And he observed that in a few seconds the leg withdrawal from the glass. This is a reaction that is called Turk's reflex. Turk's reflex. The time of Turk's reflex is about 5, maybe 3 seconds, an average duration of this reflex. And we may show the reflex arch that induces this reflex. Interconnecting a relay neuron and an efferent neuron that innervates the muscles of the frog's leg. Then Sechenov applied the crystal of sodium chloride on the thalamus, and after a while repeated the experiment with hydrochloric acid. Again he used a glass with hydrochloric acid, but he noticed that the time of the Turk's reflex prolonged significantly, or even the reflex disappeared. So it's a good example of inhibition of the reflex, and he found that this inhibition is induced by the stimulation of the thalamus. He washed out the sodium chloride crystal. He eliminated sodium chloride crystal from the thalamus, and he washed the thalamus with saline, and he repeatedly examined the Turk's reflex, and it came back. It returned, and it returned to initial time. So he proved that the recovery of reflex is possible. He didn't know, Sechenov could not find the precise mechanism, the cellular mechanism of the Turk's reflex, of his inhibition, and the reflex arch was not completely clear in that case. But he proposed that it was excitation that propagates in the central nervous system, but not inhibition. But the precise mechanism of Sechenov inhibition became clear only in 1949, and the precise mechanism we will draw here. From the thalamus, the excitation that is induced by irritation of the brain by sodium chloride descends to the spinal cord, to the segment where the reflex arch of the Turk's reflex is located. And here, this excitation is conducted to the special interconnecting inhibitory neuron that is called Renshaw cell. We will draw the Renshaw cell with the red. So the body of Renshaw cell, and an axon goes to the body of the efferent neuron. And here the GABA, the glycine. The glycine, in the case of spinal cord, glycine is released. So this is a Renshaw cell. Such interconnecting inhibitory neurons are responsible for the inhibition of reflexes in the case of thalamic of Sechenov inhibition. So actually Sechenov also proposed that the excitation propagates in the nervous system, and he did it by observing the cardiac activity of the frog. To the moment it was known that the excitation of vagus nerve inhibits the cardiac activity. So Sechenov proposed that if the excitation propagates in the central nervous system but not inhibition, then the excitation of the brain by irritation with sodium chloride will also induce the excitation of vagus. And if the excitation of vagus will be found, then the cardiac activity should be inhibited. And that was absolutely true. He noticed that after the application of sodium chloride, the cardiac activity was inhibited, and it proved an idea of Sechenov that the excitation of one brain structure may inhibit the excitation in another region of the central nervous system. Another inhibition is called Magoon inhibition. Gorazio Magoon is American scientist who found the reticular formation, very important brain structure that first is responsible for arousal, for activation, unspecific activation of brain structures, but also the stimulation of reticular formation can inhibit the reflexes by descending influences. In the case of Magoon inhibition, the stimulation of reticular formation in cat's brain could inhibit the reflexes of the bending of a leg in a cat. So this type of inhibition is also maybe called reticular inhibition due to the reticular formation. Another type of inhibition is called Goltz inhibition or linked inhibition of Goltz. In that case, we use the Turk's reflex described before. So the leg of a frog is immersed in the solution of an acid, and the withdrawal in a few seconds is observed due to the specific reflex arch. We'll draw this reflex arch now. Gray matter and interconnecting neuron and efferent neuron that innervates the muscle. But if we will simultaneously squeeze the opposite frog's leg by forceps, then the Turk's reflex will be inhibited. To illustrate the mechanism of this inhibition, we will draw the receptors, mechanoreceptors in the opposite leg, the body of the afferent neuron, interconnecting neuron. Also we may draw the efferent neuron that will contract the frog's leg muscles, but we squeeze very hard and the frog can not withdraw the leg from forceps. And from the interconnecting neuron we may illustrate the branch, we may draw the branch, the collateral of an axon, and we may draw the Renshaw cell. Also we will draw the Renshaw cell by the red color. So this is a Renshaw cell, its axon and the synapse where glycine is released, and this glycine will inhibit the motor neuron normally functioning in the Turk's reflex. So this is the mechanism of the linked inhibition of Goltz. Another example of inhibition was found by Ivan Pavlov, and this inhibition, in accordance with localization, is called cortical inhibition. In that case, we may also describe it as an orientation response when the certain reflexes, for example conditioned reflexes, are inhibited by a strange noise, for example, that stops the reflex activity and induces orientation response, arousal and orientation response. It will be a subject of discussion in the future when we will speak about the higher nervous activity. And at least we should characterize the significance of inhibition in the central nervous system. The first thing to mention is that inhibition prevents overexcitation of the nervous system that is dangerous, that may lead to excitotoxicity and to induce apoptosis and necrosis of neurons. In fact, the excitation in the central nervous system stops, the propagation of excitation stops not when the neurons became tired, fatigue, but the excitations stop when they are inhibited, actively inhibited by the specific inhibitory mechanisms. And another significant property of inhibition is that inhibition makes possible the aim-directed behavior. Then we can stop the movement of muscles precisely when it is needed to arrange the precise and skilled movement of the body. The deficit of inhibition may be observed in some pathological cases, for example, in the case of epilepsy. The severe disease with seizures when the deficit of inhibitory control of excitation weakens, decreases, and the whole brain and central nervous system became damaged by such seizures.